Thank you. Thank you very much. It's a great honor to be asked to give this kind of lecture, and it's also a challenge, asked to inspire. My goodness, it's, we will see how it works. I'm very happy to be here. So my name is Anna Karlsson. Once upon a time, I was a medical student here at the Karolinska Institute. I kind of started in 1978 or something, long time ago. I was a happy student. I liked my studies, but during the early courses, I really wanted to like deepen into something. I felt a little bit I was jumping from one subject to another, which was fine, but still I lacked something. And then at one of the early courses, someone asked if any student was interested in doing some kind of research project. And that was the right time. It was just something I've been thinking of, so I said, yes, that would be very interesting to see what it was like. And after that, I was kind of, that was the perfect combination for me. I continued my studies, but I also did research. So I had both. I have kind of continued like that, doing both, although research and university duties has become my main. I have something called the kombinationstjänst, which means that I have like 20% clinical work, which nowadays is in a clinical laboratory as a virologist working on viruses. Because that's where I started my research. Being the kind of person I am, digging deeper and deeper, I started with antiviral research. I was very much involved in HIV and chemotherapy during the 1990s and the beginning of the 1920s, and it was a very successful area. We kind of reached the goal we were aiming at, developing a good treatment for HIV infection. And during these studies, I became interested in the mitochondria and the function of the mitochondria. And as time passed by, I came into the metabolism that is very much happening in the mitochondria, the production of ATP, the production of all our building blocks for many things. I became interested in cancer cells. How do they behave in their metabolism? So the title of my talk today is related to that, the metabolism of a cancer cell. I will probably stay here for a long time in this field because there are more questions than answers still. Let me see, I didn't use this one. Oh, we forgot to, there it is. The background. Cancer cells are different from healthy cells, normal cells. And their main interest is that they promote growth, survival, proliferation, and long-term maintenance. That's their aim. Whereas healthy cells, they want to make a job. They have something to do. They have a function. But cancer cells, they want to grow and proliferate. And they have a special feature. They have an increased glucose uptake, and they produce a lot of lactate. This is special for cancer cells, and it's kind of a general phenomenon. And this was observed a long time ago. The phenomenon is known as the Warburg Effect. The Warburg Effect is defined as an increase in the rate of glucose uptake and the production of lactate. Although oxygen is present, this is what normally otherwise happens, production of lactate in our muscles if we don't have oxygen, anaerobic. But in cancer cells, they do this anyway. I have a problem with my... This is Otto Warburg. He published as early as 1926 a paper entitled The Metabolism of Tumors in the Body. So he was really an early scientist. He was working in Berlin. He was also a physician and also a scientist, studying cancer cells and compared cancer cells to normal cells. So he was the first one to identify this phenomenon. He did many contributions in the field of cancer metabolism and cell respiration. And he was awarded the Nobel Prize in 1931 for his discovery of the nature and mode of action of the respiratory enzyme. So he was really a great scientist, and the Warburg Effect has become very famous after him. So the Warburg Effect has been documented for over 90 years and extensively studied over the past 10 years with thousands of papers. It's a big field, big interest, and many proposed function. It promotes biosynthesis of macromolecules. It increases the ATP synthesis. It has effects on the tumor microenvironment, and it promotes cell signaling. All of these are suggestions, and one can see these kind of effects. But despite this intense interest and intense scientific work, the function of the Warburg effect remains partly unclear still. And the big interest in this is, of course, when the interest in studying cancer cells is to identify targets. How could we stop them being cancer cells? That's why one wants to understand the details about how they promote their life. So this is a figure of the papers. You can see that from the beginning there was not so much activity in this area, but for the last 10 to 15 years, the amount of papers and the amount of publications has increased very, very much. On the Warburg effect, on ATP, this is on the Warburg effect, and biosynthesis and the Warburg effect, ROS production and the Warburg effect, acidification and the Warburg effect, acetylation and the Warburg effect. So the frequency of publications is dramatically increasing. So, to our work, if I had had another audience, more specialized, the title of my talk could have been like this: Cancer cells control the redox balance in glycolytic metabolism through regulation of glutamic oxaloacetate transaminase 1. That title I could have used. But one can always simplify. Studies of GOT1 is what I'm going to tell you about. What is this GOT1 and why GOT1? That's always a very good question. Why do you study this? Why do you investigate this? And we had a previous study. That is what normally happens. One studies something, and you get answers to what you're studying, and you can kind of report your results. But meanwhile, you also find some other things, some other unexpected, like things that you did not, and that was not really the focus of your first study. So that will become your next study to investigate what was this that we saw. So that's kind of how the chain of work continues very often. We became interested in this enzyme here, converting aspartate to oxaloacetate, very close to the central metabolism the reason what we observed was from a previous paper where we had studied a previous work. We had studied I told you I was interested in the mitochondria. We are interested, and then we were interested in the transport between the cytosol and the mitochondria. And our interest was focused on, or we became interested in this carrier here because it was upregulated in many cancers. They had more of this, so we were investigating the role of this. And we could see that if we shut this off, the cells became less malignant. So this could then be a good target to develop something that could have effects on cancer, because this was identified. If you block this carrier, the cells become less malignant. While while doing this investigation, we found a decrease in these mutated cell lines of this enzyme, the GOT1, which was not really part of our focus at that time, but it was an interesting finding, the decreased expression of this GOT1, because it was so pronounced in our new cells that we made. So for us, it was quite obvious for the next study was, what was this? Why was this seem to have a regulatory function? Why? And what was the, what did it do? So this is the way we address questions. We mutated it. We disrupted the gene for this enzyme in a cancer cell line. And today they are quite nice tools, some CRISPR-Cas9 methodology that we use to make a frame shift so that you could kind of, you got rid of there was no open reading frame anymore. There was no protein, so it was really a zero mutant. Then of course we investigated the cells in many, many ways. But the main message was that these cells, the red ones are the mutated cells, they were growing as the wild type. One always compare with a normal cell. So the normal cell was growing like this. The mutated cell was growing for a couple of days, and then it stopped growing, and then it stopped growing. So there was clearly something that had to do with it needed to be happy growing. It needed this GOT1. We, after many gene expression analysis, trying to substitute different compounds in the cell medium, we identified that these cells became very heavily depending on glucose. They did not grow if glucose was going down. And this was always in comparison with the wild type cells, of course. So glucose was something that was crucial to these cells. And this increased glucose dependency that we could also measure, of course, we had all kinds of indirect signs, but we could also measure it. We could see that it was increased in the consumption of glucose from the cell medium in these mutated cells. And this is our conclusion of this study. The cancer cells, as we said from the beginning, they consume a lot of glucose. That is the Warburg Effect note. And then they make, metabolizing, they will produce a lot of NADH. That is a compound participating in many, many chemical reactions, and it's a redox active compound. And to balance this production of NADH in glycolysis, one needs to produce the other in this pair is the NADH, and to do that, pyruvate is converted to lactate, and that will produce the NAD+. So when disrupting this GOT1, the gluconeogenesis that produces glucose, so there are two ways of getting glucose for the cells, either externally, uptake from the cell medium, or from the gluconeogenesis from the cell metabolism. And it turned out that GOT1 inhibit this pathway of producing glucose. So these cells with a decreased or absence of this enzyme, they became completely depending on the extracellular glucose. And to produce the lactate and to produce the NAD+, that needs to keep the redox balance in the cells. So that was our conclusion from many, many studies, of course. And it turns out that when glucose is low, this is high, and when glucose is high, this is low. So this is really a balance to counteract the medium and the availability of glucose that the cell needs to survive. And that is why the cells grow normally for the first 3 days, my first picture, because then there was a lot of glucose in the medium, but these mutated cells. But after 3 days, they stopped growing because the mutated cell could not produce their own glucose, which the wild-type cells could. So that was the conclusion we made. Because most of all, the cells, even the cancer cells, they want to grow, they are selfish, they don't, they do what they want, but they still need to keep the major features to be alive, because they want to be alive, of course. And the very important things for the cell machinery is to keep their redox balance. So they need to have this functioning, otherwise they die. If we continue to grow these mutant cells without glucose, they will die immediately. They have no other way of surviving. So it's a very explosive way when one looks at it in a microscope. It is within minutes, because they are completely out of any alternative way of feeding themselves. And of course, this is a good way for us to start thinking that we have found a very sensitive spot for the cancer cells where they really have no way out to survive if we can hit these pathways. So to summarize this work that is really ongoing, it's a project we have running right now, is that the gluconeogenesis to make its own glucose is crucial to counteract the glycolysis of cancer cells when glucose concentration is low. The cancer cells need to have this glucose. And that this enzyme that we identified is regulating this pathway, that the GOT1 is a principal source of oxaloacetate for this gluconeogenesis when glucose is low during glucose deprivation of cells. And of course, sometimes tumors have a glucose deprivation. They don't have high nutrients all the time. So then this enzyme goes up, and if they have high nutrients, it goes down. So it has a regulatory function. So we conclude that this enzyme, it regulates the redox homeostasis through generation of NAD+ from the gluconeogenesis to keep the balance in the cell. So this is my group. It's Shaoshan Zhu who has mainly done this work. As you know, we professors, we don't do much lab work ourselves. Nowadays, I used to like doing lab work. I did for many years, but not nowadays. And then there are postdocs and PhD students. Some postdocs have left now, and PhD student Shuba has left also. And we have new. But that's how a research group look like, and that's how we work. Thank you for your attention. A difficult topic for questions, but any questions is of course welcome. Yes?
Hello. I'm thinking about your cell line, your cell
yes.
lines. Do you get the, if I understand right, there's a number of cancer cell lines. The first question is, does your cell line represent a particular kind of cancer? And do you get the same effect in other cell lines, other cancer cell lines? Do you see the same phenomenon?
I got a question about other cell lines. How many cell lines did we use? We used osteosarcoma cell line. We have used lung cancer cell lines. We have this we have done in 3 different cell lines, because your question about the cell line is of course very relevant. You don't want to study something that is so particular that it's only valid for one cell line. So we have studied 3 different cell lines and seen the same thing. But of course this is still cell lines, and of course to publish this work we probably need to do some in vivo model of our findings. That's what they request nowadays. Because a cell line is always a cell line. But it's usually, my experience is that cell lines is a good model because you can control it quite well. You compare a wild type cell line with a mutant cell line. So you kind of have the same, otherwise genetic background. But that's always one always have to be aware. I'm studying a cell line, a cell line of cancer. I'm not studying a patient's cancer. So, yes?
When you screened the compound that may impair GOP1, you selected the glucose at last. What other compounds did you screen?
Oh, we didn't screen any compounds. This was a mutant. We inserted a mutant. But it would be nice to have a compound. Yeah, screening for compounds would be a very interesting thing to do. That's a very good idea.
Then how did you decide to study glucose?
We did many experiments. First of all, in a gene array, when we looked at different, we saw that we saw that there was upregulating of some glucose genes in the glucose metabolism. So we focused on that. We added different compounds to the medium that were upstream or downstream, and we could see what could rescue and it was only glucose that could rescue the phenotype. After all, we tried lots of different compounds, and it was only glucose that could rescue the phenotype, and some compounds upstream of this mutant. Yeah. So it was a little bit like a puzzle. You need to rule out what it could be.
You mean in the figure you showed upstream of the GOP1?
Yes. Yes. We could not rescue with downstream. We could rescue with upstream and glucose. So we kind of pointed that to be this, and then we had an hypothesis, and then we tested the hypothesis.
Which type of cell lines did you use? I didn't hear.
No, this was osteosarcoma cell lines, osteosarcoma cell lines. But we also tested in some some lung cancer cell lines, and we had 3 different cell lines we used. Okay, thank you very much.